Good day, everyone. Today we are going to discuss about genomics and proteomics. But first, we are going to discuss what is all about genomics. Genomics. It is the study of entire genomes, including the complete set of genes, their nucleotide sequence and organization, and their interactions within a species and with other species. Genome refers to an organism's entire genetic makeup. Now let's differentiate genetics and genomics. When we say genetics, it is the study of heredity, while genomics is the study of entirety of an organism's genes. Gene refers to a specific sequence of DNA on a single chromosome, while genome refers to an organism's entire genetic makeup. In genetics, it involves the study of functions and composition of the single gene, while genomics addresses all the genes and their interrelationships. Genome mapping. Genome mapping is the process of finding the location of genes on each chromosome. The maps that are created are comparable to the maps that we use to navigate streets. A genetic map is an illustration that lists genes and their location on a chromosome. The genetic marker tends to be inherited with the gene of interest, and one measure of distance between them is the recombination frequency during meiosis. Early geneticists called this linkage analysis. Physical maps get into the intimate details of smaller regions of the chromosomes, which is similar to a detailed road map. A physical map is a representation of physical distance in nucleotides between genes or genetic markers. The main goal of genomics is to sequence the entire genome by cutting it into small, manageable pieces. Assemble the entire genome from the pieces and understand how gene expression takes place. There are three subfields of genomics. These are structural genomics, functional genomics, and comparative genomics. Now let's move to genome sequencing. During genomic sequencing, the first step is genomic DNA, which is first extracted from a sample cell of an organism and then is broken it into random fragments. Next, fragment cloned in DNA or the carrier that is capable of carrying large DNA insert. And lastly, the cloned DNA is then extracted from the bacterial clones and is fed into the sequencing. The whole genome sequencing is a process that determines the DNA sequence of an entire genome. Whole genome sequencing is a brute force approach to problem solving when there is a genetic basis at the core of the disease. The first human genome sequence was published in 2003. There are two approaches in genome sequencing. First is the hierarchical shotgun sequencing and whole genome shotgun sequencing. When we say hierarchical shotgun sequencing, it is useful for sequencing genomes of higher vertebrates that contain repetitive sequences. And whole genome shotgun sequencing is useful for smaller genomes. Next is the Human Genome Project. The Human Genome Project is an international scientific research project with the goal of determining the sequence of chemical base which make up human DNA, and of identifying and mapping all the genes of the human genome from both physical and functional standpoint. Human Genome Project was formally founded in 1990 by the U.S. Department of Energy and the National Institute of Health and was declared completed on 14th day of April 2003. The basic goals of Human Genome Project is to identify the genes and their functions in human DNA, to determine the sequence of 3 million base pairs that make up the human DNA, to develop tools for data analysis, to obtain physical map of human genome, and to store the information in public databases. Benefits and applications of genomics. Genomics can be used in the field of medicine for early detection of genetic diseases and its diagnosis and treatment. It is also useful in the field of agriculture, also to study evolution through mutation lineages, and also useful in forensic science. Now that we've already discussed genomics, let's move to proteomics. Proteomics is the study of entire set of proteins expressed by a given type of cell under environmental conditions. In a multicellular organism, different cell types will have different proteomes, and these will vary with changes in the environment. Unlike a genome, a proteome is dynamic and under constant flux, which make it more complicated and more useful than the knowledge of genomes alone. And the entire set of proteins produced by a cell type is called proteomes. Proteomes can be studied using the knowledge of genomes because genes code for mRNAs and the mRNAs encode proteins. Proteins are the final product of genes that perform the function encoded by the gene. Proteins are composed of amino acids and play important roles in cells. All enzymes except ribozymes are proteins and act as catalysts that affect the rate of reactions. Proteins also are regulatory molecules and some are hormones. Transport proteins such as hemoglobin help transport oxygen to various organs. Antibodies that defend against foreign particles are also proteins. In the disease state, protein function can be impaired because of changes at the genetic level or because of the direct impact on a specific protein. There are several basic techniques in protein analysis. First is mass spectrometry. Mass spectrometry used to identify and determine the characteristics of molecule. Next is X-ray crystallography. X-ray crystallography enables scientists to determine type of three-dimensional structure of a protein crystal at atomic resolution. Next is NMR, or the nuclear magnetic resonance. It uses magnetic properties of atoms to determine three-dimensional structure of proteins. Next is the protein microarrays and the two hybrid screening. When we say protein microarrays, it is used to study interaction between proteins, and two hybrid screening is used to determine whether the two proteins interact. Next is the Western blot or protein immunoblot. This is a technique that combines protein electrophoresis and antibodies to detect proteins in a sample. Next is the cancer proteomics. Cancer proteomics encompasses identification and quantitative analysis of differentially expressed proteins relative to health tissue counterpart at different stages of diseases, from preneoplasia to neoplasia. Genomes and proteomes of patients suffering from specific disease are being studied to understand the genetic basis of the disease. The most prominent disease being studied with proteomic approaches is cancer. Proteomic approaches are being used to improve the screening and early detection of cancer. This is achieved by identifying proteins whose expression is affected by the disease process. Biomarker and protein signature. An individual protein is called biomarker, whereas a set of protein with altered expression level is called protein signature. For a biomarker or protein signature to be useful as a candidate for early screening and detection of a cancer, it must be secreted in body fluids such as sweat, blood or urine so that large-scale screenings can be performed in a non-invasive fashion. In cancer research and medicine, biomarkers are used in three primary ways: the diagnostic, prognostic, and predictive. Diagnostic, identifying early stage of cancers, while prognostic is to forecast how aggressive a condition is, and predictive is how well a patient responds to treatment. Now, the current problem with using biomarkers for early detection of cancer is the high rate of false negative results. When we say false negative results, it is a negative test result that should be positive. In other words, many cases of cancer go undetected, which makes biomarkers unreliable. Some examples of protein biomarkers used in cancer detection are CA125 for ovarian cancer and PSA for prostate cancer. Protein signature may be more reliable than biomarkers to detect cancer cells. The National Cancer Institute has developed programs to improve the detection and treatment of cancer. The Clinical Proteomic Technologies for Cancer and the Early Detection Research Network are efforts to identify protein signatures to different types of cancers. The Biomedical Proteomics program is designed to identify protein signatures and design effective therapies for cancer patients.